directionality affects the structure, structure affects the function. Okay, so there's two of them. Here's the first one, nucleic acids. So if you go back to where we were talking monomers, right? Mono means one, mers, roughly unit, 1 unit. Monomers are linked together to make the polymer. Well, remember, if you go back and think about it, there are two kinds of nucleic acids. One is RNA, which expresses the instructions from the DNA. The DNA is the second one. It stores those instructions, that information. And if you look real close, and if I were you, I would probably draw that and label this. This is a monomer. This is called a nucleotide. This is a nucleotide of nucleic acid. Okay, so nucleotide, right? It's a monomer. So if you put a lot of these together, they're going to arrange themselves in a certain way. And the way they're arranged is very directional. And you're going to notice here's 3 parts. I think this is the kind of a review. So here's your phosphate group, and that was kind of easy to see. You got that phosphate surrounded by a bunch of oxygens. Okay, so you got a phosphate group. You have here in the center, you have a 5-carbon sugar. It's either going to be ribose or it's going to be deoxyribose. It's going to be ribose or deoxyribose, depending on what you're talking about, DNA or RNA. And then over here, you have what's considered a nitrogen base. Now, I'm not going to write it down. You need to write it down. And that could be one of 4 things. If it's DNA, it's going to be adenine, thymine, because A always goes with T in DNA. And then you got C and G, cytosine and guanine. Those are the 4 choices you could have for DNA. In RNA, though, the T is gone and it's replaced with uracil, a U. Remember, RNA is expendable. It's cheap to make. The body can get rid of it, but you can't get rid of your DNA. So it never uses DNA in this case. It's always going to use RNA to express. That's why. Okay. Now you're going to see this right here. 1', 2', 3', 4', 5'. Remember, this is a 5-carbon sugar. And why they call it prime, I don't know. But whenever you see a diagram like this and you don't see anything here, but you see all the other elements, that means there's probably a carbon there. Okay. Especially we're talking OChem, organic chem. You're going to see a carbon. So there's going to be 1 carbon, that's the 1' carbon, 2' carbon, 3, 4', and the 5'. Okay. So make sure you get that down. Now, DNA, RNA, is arranged in this 5'. You're going to see it. It's going to be a 5' to 3'. So another one's going to attach here. It's going to be a 5' down here. You see what we're getting at? Think of this. This is 1 strand of DNA on one side. This is before it coils up. Here's the opposite strand. Look what they're doing. If my hand shows the direction, this is how they're arranged. They're never arranged like this because it doesn't match up. It's going to be 5' to 3'. Okay, so let's say 5' to 3'. So it runs anti-parallel. That's the way they're called. Anti-parallel. And so whenever you see this, remember, it always comes into, well, this comes into play, especially when we start talking about DNA replication. But very directional. That's that directionality we're talking about. Okay, here's a picture of a DNA strand. So if you look at it, you can pick out the nucleotides. Here's 1. Here's the 5'. Up here, 3'. 5, 3. And then if you notice the other side, can you see something odd there compared to this side? What's it look like? Think about it. It's opposite, isn't it? It's just the opposite. So you have your 5, and you can see there's the 3, there's the 5. So it's the directionality there. Okay, so the arrangements of the nucleotides are going to dictate the instructions given, right? So Do you think my DNA is exactly like yours? No. Is it close? Oh yeah, very, very close. What about you compare my DNA with, say, like a cat? Close? Probably not. Okay. The closer it is, the more of the species. And so there's the lady holding her arms that way. And so nucleotides, always in a linear sequence. That means they're always in a line. So you got one half of the DNA is always linear. It's in a line. And it's got to be in a certain order. Remember, this is like a code. I want you to think of DNA as a code. And it has to be written in a certain way if you want to get a certain outcome. In the case of human beings, it's got to be written in a certain way. If you want a cat, you got to write it in another certain way. Okay? So DNA, anti-parallel, and they're held together right here by covalent bonds. Biological molecules love covalent bonds. Covalent bonds. Now, one thing I want you to notice, it's kind of hard to see, but right here I got the number 2 written because there is what's called a double covalent bond there. Remember, A's will not go with C's or G's. This is why. Here's the C and the G. Look here. Here's a, oh, I'm sorry. I told you wrong. This is hydrogen. And these are hydrogen. These are covalent out here. Okay, so there's a triple bond. Very, very specific. Okay, any questions? All right. Okay. Proteins. We're going to become experts, semi-experts on this. Okay, so whenever you see polypeptide, they're just talking about a protein strand. So up there in the top right, you see polypeptide 1, polypeptide 2. Look, linear structure, same thing, linear structure. Proteins are made up of monomers. Do you remember what they're called? There's 20 of them. Amino acids. And they've got to be in a certain order for a certain protein to work. And so you got 2 totally different ones. Okay? So you see the line, you see that's different, it's definitely directional. And there's bonds that hold it here. These are called peptide bonds. Those are covalent bonds. Peptide bonds. And again, peptide is just a fancy word for protein. And so you can see all the covalent bonds. You lose a water molecule, they attach. You add a water molecule, these protein strands will break apart. You take out a water molecule, they attach. You add water, they break apart. Okay? Now 4 structures for proteins you got to be familiar with. You're actually going to mess with this today. And you kind of probably would be wise to put it to memory. It's not that hard. Primary. Just think of primary as the first. So at the most simple level, this is what our protein starts. It's just a string of amino acids in a line in a certain order. That's all it is. That's called the primary. First level or primary level, first structure. Secondary is where the amino acids here interact with each other. Not with anything else. They just interact with each other. You're going to see this today as we start cutting out our paper protein strand. Okay? And then the tertiary is the completed protein. This is your actual, after this is all done folding, this is going to be your 3D shape of your protein. So after secondary is all done folding, you're going to have this. Now, for those of you that like this stuff, they can either take, in the secondary, they can take what's called an alpha helix shape. So it's just going to be like this. It's an alpha helix. I'll write it down. And over here, you're going to see it looks like somebody's folded paper and folded paper and folded paper. These are called beta pleated sheets. So basically, when they start folding, they're going to fold into either of those two shapes, and then they're going to keep interacting until they are finished and connected into this 3D shape here. Now, quaternary. Now you notice how all those words have a play on the letter or the numbers, right? Secondary, tertiary is 3, quaternary is 4. This is interactions with other proteins. Interactions with other proteins. Let's add 3D proteins. Okay, so in this picture, normally there's 4 of them here. You can see, but here they're only showing you 2. So here's one completed protein, and then you see this darker blue. That's another one. Okay. That's the completed protein strand. And remember, if a protein is not folded in the right way, it is useless. It will not work. Okay, so primary, secondary, tertiary, quaternary. Quaternary is the very last. That's it. All right. And there's kind of a different view of it. You can check it out. I'll leave it for you. You see it's folding up. There's a 3D. There's another, probably a cooler picture. Okay. Now, last one. Carbs. Again, structure always is going to determine the function. But I think here what they want you to know, remember carbohydrates, remember that's our #1 energy source, 60% of our diet, carbohydrates. They're going to form linear chains of sugar monomers. So if I would blow this up, here you have, sorry about the drawing, you have a glucose, and you see how the glucoses are attached together. There could be some others in there, but you can see the top 2 are plants. Cellulose, look how tough it is. It's really long, it's stacked. That's why we can't digest it. And then starch, not quite so intricate and stacked, but you can see it's all, they look very similar for animals, but you can see these are called branched chains. Branched chains, and they're held together right there. These are called glyco for glycogen. Glycosidic bonds, which are covalent bonds. It's just another type. So for you guys, you would need to know peptide bond, covalent bond, and glycosidic bonds. That's another covalent bond. And this is always the giveaway. Always the giveaway. I never knew this. Glycogen, carbohydrates. All right. I think I'm finished. We only have one more short bit of notes. Can't remember. We're going to make cheese Friday. I wasn't sure I could get all the ingredients.